bio students. Hope you enjoyed your long weekend. Today we are going to talk about the second stage of protein synthesis, transcription. Last week we talked about the first stage, translation. Translation helps us to make a protein. Translation in review, or transcription in review, is transcription is when a gene in DNA is copied into messenger RNA. The messenger RNA then moves from the nucleus with the copy of the code to the cytoplasm where a protein will be made. The second step of making a protein is stage 2, translation. Translation is using messenger RNA instructions to make a protein. This occurs on a ribosome that's located in the cytoplasm. When you take a foreign language, you often have to translate that language from English to Spanish or vice versa. A genetic translation changes RNA language into protein language. Remember that DNA holds the instructions for making proteins, and those proteins give us our characteristics. How do we read RNA? RNA is read in a three-letter word called a codon. One codon in RNA language equals one amino acid in protein language. Therefore, several codons are going to equal several amino acids joined together, which is going to be a protein. Now, there's a code that we use in order to read these codons. A codon is going to be translated into an amino acid. So there are several ways in which we can translate the key from messenger RNA to the protein language. And one way is to use a codon chart, like you see here on this slide. So let's pretend that we are going to use this codon chart to look up a codon. Let's look for the codon AUG. So if we use this chart, the largest letters in the center here are the ones that would be the first letter of the codon. So A is our first letter of our codon, so there is A. Now, within this portion of the chart then, we're going to stay in this portion. So our second letter is U. So U is the next largest. The second letters are the next largest letters. And then G, the third letter of the codon, is the smallest letters, A, U, G. That codes for the amino acid methionine. So we would write methionine, or sometimes scientists just abbreviate by using the first 3 or 4 letters. So you can use the codon chart in this way, or there's another option. This is your second option for your codon chart. Again, same thing, except you just read this chart in order. First letter, A. Second letter, U. So that means that our answer has to be within this section of the chart. Third letter, G. So we go across, and our answer again is methionine, or start. You decide whichever chart you want to use, whichever makes more sense to you. There is always a start codon at the beginning of the messenger RNA chain, and the start codon is always AUG. There is also always a stop codon at the end of the chain, but there are 3 possibilities for stop codons: UAA, UAG, and UGA. The key concept to remember is translation converts the messenger RNA message into a polypeptide or protein. So let's practice using the codon charts. You decide which codon chart you want to use. So we already know that AUG codes for methionine. So we know the answer there, and it's start. Now I want you to stop the video and try to use your codon chart to translate the rest of the message. Now, let's check your answers. GUC codes for valine. AGA translates into arginine. CCC, proline. And UGA, stop. Hopefully you guys got them all correct. Let's practice some more. Again, AUG is methionine. AUC codes for isoleucine. GAU, aspartic acid. CCG, proline. And UAA, stop. If you have further questions, you guys can ask me when I see you in class. So in review, translation is the process that uses the message of mRNA to produce a protein. The messenger RNA is read in a 3-letter word called a codon, and the transfer RNA has an anticodon, which are complementary to the messenger RNA and allows the transfer RNA to carry the correct amino acid to the ribosome. So on this slide, there is a link for a little animation that shows you the whole process of protein synthesis. When you're done with this, or if you want to stop now and watch the animation, you can click and open on this link, but make sure that you use the Firefox browser or it will not work. So the first step in translation is called initiation, which would be at the beginning. At the 5' end of the messenger RNA strand, it is going to bind to the ribosome. The second part of this is that the first transfer RNA will be carrying methionine, which is going to enter the ribosome and bind to the start codon, AUG. Elongation is the next step in translation. This happens when another transfer RNA is going to enter and bind to the second codon. So this anticodon on the transfer RNA is going to bind and be the complement to the second codon. The ribosome will then join the amino acids together right here with a peptide bond. Transfer RNAs continue to carry in and drop off amino acids as the polypeptide chain grows. Termination is the last step. A stop codon on the messenger RNA is read, and the polypeptide then will detach from the ribosome. So this protein is going to come off of the ribosome when it is done, so that it can be used in the body or the cell wherever it is needed. Remember that transcription makes 3 kinds of RNA: messenger RNA, transfer RNA, and ribosomal RNA. On one of the ends, transfer RNA molecules are going to carry amino acids. There are 20 different amino acids. On the other end of the transfer RNA, we have an anticodon. This anticodon is going to be complementary to the messenger RNA strand. Your example, the messenger RNA codon ACA and the transfer RNA anticodon UGU will allow the transfer RNA to bond to the messenger RNA and transfer the correct amino acids in the growing protein chain. Here's just an example of the process. So let's have an overview of the 3 different things that we have been talking about in this unit: DNA replication, transcription, and translation. So DNA replication is when you use DNA to make DNA. And this occurs in the nucleus. When we use DNA to make RNA, that could be called transcription, which also occurs in the nucleus. And then when we make RNA to make a protein, that is called translation, which occurs in the cytoplasm on a ribosome. So in your packet, let's do some practice, going from DNA to RNA and then to making a protein. So using your codon chart, not yet using your codon chart, but take the DNA here, TAC, and then you are going to transfer it into the RNA. So remember that RNA does not have a T, it has U instead. So T always goes with A goes with U, C goes with G. C goes with G, G goes with C, G goes with C. Okay, you try the next one. The answer should be AUU, GCA, and UGA. Now, you want to take this RNA, you want to take the RNA codons, you want to use your codon chart, whichever one you prefer, the circle or the chart, and you want to translate this messenger RNA into the protein language, which amino acid would code. Stop the video and practice your codons. Okay, as we know, AUG codes for methionine, or the start codon. GCC is alanine, AUU is isoleucine, GCA is alanine, and UGA is stop. Try practice problem number 2. Remember, there is no T in RNA. Using your codon chart then, translate the RNA into the protein language of amino acids. Methionine, tyrosine, arginine, glycine, and stop. Oops. Okay, now you're going to watch this video and complete the other side of your video worksheet. Lastly, your homework is on page 23 of the packet. Please write down any questions that you have, and I'll be happy to answer them when we see each other again. Thanks.